I did my research on understanding the actions of membrane binding proteins. Mainly, we focused on the protein E sites, and we did this through optical tweezers and liposome co-sedimentation assay. My name is Swetha Somasundaram. I'm a senior at Arcadia High School here in California, and yeah. Mainly, I'm going to go a little bit into explaining the research, and then I'll show a couple of pictures that will help guide what I've written here. Membrane binding proteins are mainly just exactly what they're called. They bind to membranes. Proteins have multiple domains. E sites was a specific protein type that is calcium dependent and has multiple C2 domains. We use techniques involving newer technology. We use optical tweezers, which is something that's a new technology that's sort of rising right now. We used faster centrifuges for liposome co-sedimentation assay, which is a new type of technique used to understand supernatant in pellets. And mainly our research was to understand how membrane proteins bind and what causes them to bind. During a meeting with my mentor, we asked the question, is calcium that is being used to bind to these proteins to the membrane, is it quantitative? Is it a co-inhibitor, or does it actually just help and does it get used up within the binding of a protein? And we did a separate experiment to test this hypothesis. So our research was a little bit more expanded than we had expected it to be. Here's a picture of E sites. Part A shows what E sites actually is. I'm going to show right here. This is the E sites that we focused on, E sites 1. There is an E sites 2, but we did not focus on that as much as we did the E sites 1. As you can see, the calcium is the red dot right here. And the calciums are over here as well. There are 5 C2 domains on the E sites. C2E is not calcium dependent, so that can bind to the membrane without any help. C2A and C2C are calcium dependent. Even though B and D are not calcium dependent, they are linked to A and C, so they are forced to only bind when A and C are willing to bind as well. So calcium is very much necessary in this scenario. Part B, also shows how the optical tweezers helped us analyze how these C2 domains bind to the membrane. So this right here is your double bilayer membrane. And then you have this right here is a polyestyrene bead. So a polyestyrene bead is placed onto a section of the protein, and then you start to pull it out. So a lot of it just looks like you have the protein here, and then you're starting to pull it out from that membrane. We pull out the C2E binding first. Once we pull that out, you can pull out the C2D and C, and then C2A and B, and that helps us notice how the protein binds to the membrane, how that calcium really helps it bind and link onto the membrane, and then pulling it out also shows us the way and the movement of the protein, how it folds when it binds onto that membrane. Right here is how an optical tweezer works. You can see that there's a laser that comes onto the bead, and that bead kind of just traps the section that we are looking at and that we're trying to open and close. And so, yeah, you can see that there's a voltage that's necessary. We need to give it some electricity, some energy to actually start moving it around. This right here also showcases the force that was necessary and how we pulled it out. So we have 2 separate little optical tweezers that are going into it. We have one that's holding it down and the other one that's pulling it out. So a lot of it is minuscule just movement to see it on see the protein binding and unbinding in real time instead of that super quick movement that it has in our bodies. And this just shows the differences in the binding. When it's up here, it means that it has been binded to the membrane, and then when you take it off, it goes down to here. So it really just showcases how each domain looks when you pull it off and when you put it back on, and the force that's necessary to do that. This was the co-sedimentation assay that we did. We took 3 separate centrifuges and really looked at it that way. We did 3 separate experiments. We did it with the liposome, which is the thing that we're using as a membrane, and the calcium, without the calcium, and then just the liposome and calcium together. So being able to see that difference in all of this was really, really interesting. A lot of it was here without the calcium. You have just the liposomes down here, the pellets, a couple of protein binded, and that was because of the C2E domains that are non-calcium necessary. And so they were able to bind to the liposome itself. And then the supernatant right here, so all of that protein is still up on the top. It did not really bind with any of the membranes. Here, when calcium was added to it, everything became a pellet. There was no supernatant left over. There was no liposome that wasn't binded to something. Everything was binded together. Everything was great. And then we did a separate section just to ensure that the centrifuge was working properly, to see if the pellet and the supernatant were on there properly. And then, as you can see, these are the results. Right here, we have the calcium and the liposome together. You can see that the pellet was highly, highly favored when we had both calcium and the liposome added together. When there was no calcium, the supernatant was favored. So we had a lot more of that protein left over and nothing really binded together. So this really showcases how calcium is necessary, but not quantitative. Calcium is just there and then can be taken off when the protein is done working on the membrane. It can be unbinded and the calcium just kind of pops off of the protein in itself. Working with my mentor was really, really great. She took me on a little lab tour. She was very, very knowledgeable about her work. She was doing her postdoctoral associate work right now. And she was able to just show me around her Yale lab. The pictures that I've posted here are the optical tweezers in itself. This is the little sections that kind of just have the lenses. So those are the lights that we can use to look through the microscope to see them. This is the mirror that I showed in the previous picture of the optical tweezer, and it kind of just like pushes in onto the bead and really ensures that we can move that around properly and in very minuscule movements. She gave me really, really good advice, whether it be about the research in itself or about what I should do in the future. Cell and molecular biology is something that's very important to me. So whether or not I go into protein understanding, she did give me a lot of insight into how to work on cellular research. She also gave me a lot of her various research articles and a lot of the research articles that her lab has been working on in previous years. And she also showed and helped me meet with her main professor and her main owner of the lab. So it was really nice just to get those connections and be able to talk to other people that were very, very knowledgeable about their work in this field. My reflection on this, I felt a lot stronger about my future plans and what I want to do in the future. My interests, like I said, are in cell and molecular biology. So being able to use those new technology ideas that she gave me, optical tweezers, liposome co-segmentation assay, and to put that into practice in cell and molecular biology, mainly in diseases like cancer or cystic fibrosis, things like that. Being able to do that helped me just feel stronger about those future plans that I might have for myself. It did help me gain experience, even though I've taken AP courses in science and done labs and that. It wasn't as interesting as this research experience and being able to actually hands-on write an analysis and a research paper on my own. It also gave me chances to ask questions and make progress in research with my mentor and be able to actually understand what research is in a main medical field, in the science field, to just understand what research really is and how it works. And being able to help my mentor in making those connections. She also let me know that I could help her with future research. The questions that we asked and the further progress and the extensions we did in this research analysis helped her just understand what she wanted to do for her future research. So she said that I could help her out with that. And questions that we asked during our sessions kind of just gave both of us ideas for what we wanted to do in the future and her ideas on what her lab could be doing in the future. Lastly, I want to thank the ATHENA by Women in STEM organization and the Talaria team for this amazing opportunity. Thank you.